Hi, everybody. Welcome to our Del Norte iGEM lung cancer workshop, where we'll be discussing lung cancer in general and also how we will be using synthetic biology in this year's project to address lung cancer and create and come up with a diagnostic tool to help with early diagnosis for lung cancer. So let's jump right in. So I'm going to introduce myself. My name is Anya Sabhnani, and I am a junior, and this is my second year in Del Norte's iGEM team. And I'll just answer one of these questions below. My favorite ice cream flavor is cookie dough, and it would be great if each of you could go around and answer this in the chat. Awesome. So we just wanted to start off with some statistics so that you can really understand how prevalent of an issue lung cancer really is and how important early diagnosis is to the issue. So 1 in 2 men and 1 in 3 women will experience cancer in their lifetime. Cancer afflicts 1.7 million Americans each year and kills 600,000 of them. So you can see that there's a high percentage death rate. And 40% of cancer cases have no known cause at all. Cancer mortality has been slowly declining, down 20% over the last 2 decades. And if society were to implement what's known about prevention, cancer deaths could be reduced by 50%. So how does knowledge about cancer make it less scary and lead to new and effective treatments? Well, personally, I think that knowledge about cancer lets us understand what is required to help and what goes on in the body and what we can do to sort of deal with that and treat that. So how do you guys think that cancer happens and how does it affect people who have it? These are some questions that you should think about as we go through the workshop because we'll return to it at the end. And where does cancer come from? How is a cancer cell different than a normal cell? And how has cancer treatment evolved as scientists have learned more about cancer? And what steps can an individual take to prevent cancer? And why has it been so difficult to find a cure for cancer? All of these questions will be addressed in this workshop, and it's really important that you keep them in your mind so that you fully understand this issue. Okay, so before we really dive into the synthetic biology aspect of our project, here is some background. Let's start from the basics, something that you have all seen before. DNA, or deoxyribonucleic bases, are guanine, cytosine, thymine, adenine, and they're labeled on the photo so you can see complementary base pairs. So guanine and cytosine are complementary, and thymine, adenine are complementary base pairs. Okay, and so these make up the sugar phosphate backbone of DNA, and that's what makes the double helix. Very simple. And things. So leading into cancer, so an initial mutation has a negative cell cycle regulator. So like cancer cells are results of these mutations. So mutation overactivates a positive cell cycle regulator. And then a third mutation inactivates a genome stability factor. So all of these are like important things to like the normal cell cycle that these mutations are changing to create this cancer cell, if that makes sense. And then additional mutations accumulate rapidly until a cancer cell is created. So that goes back to our previous question of how cancer cells differ from normal cells in the human body and how they can potentially be dangerous. So this brings us to the question, what exactly is cancer? So cancer begins with uncontrolled cell growth, and cell growth meaning the cells that were addressed in the previous slide. These cells attack the body parts, or it's called malignance, and you are made up of trillions of cells that over your lifetime normally grow and divide as needed. But when cells are abnormal or get old, they usually die. Cancers start when something goes wrong in this process, like the mutations. Cells keep making new cells, and the old abnormal ones don't die when they should. So thus the growing and acceleration of growth of these cancer cells as they continue to reproduce. As the cancer cells grow out of control, they can crowd out normal cells. This makes it hard for your body to work the way it should. And like a group of cancer cells is called tumor, a term that you guys are probably familiar with. And these tumors can form in your body in multiple places, one place. It depends on type of cancer. And that's what ultimately lowers your bodily functions and can lead to fatal symptoms, side effects, and eventually death. Okay, let's move on. So this brings us to the discussion of genes and how people inherit certain characteristics from their parents. So genes are the instructions for organisms, and they consist of DNA and are stored in the chromosomes, which we each have 23 of. And like books with 4-letter combos and triplets. So like 4-letter combos, it's like kind of like the like DNA, like I mentioned earlier, has the complementary bases adenine, thymine, cytosine, guanine, and those like 4-letter combos A, C, G, and T are like repeated over and over again. So in every living thing, from bacteria to lettuceheads to humans, there is DNA and they have genes. Traits result from DNA transcription or the copying and translation of DNA. And translation of DNA is when it changes from RNA or ribonucleic acid to proteins. And so here's a little diagram to sort of explain that. So your body is made up of cells, and then going down to the simpler, or it's cells, and then in cells there's chromosomes, and in chromosomes there's DNA, and DNA makes up genes, or is made up of genes, sorry. And as you can see in this chromosome sort of diagram, you can see the sort of double helix structure of DNA running through it. This hopefully helps you understand how everything in our body is so interrelated and interconnected, and and genes and DNA really are the building blocks. Okay, move on. So as mentioned earlier, there's DNA translation, which is RNA to proteins. So what exactly are these proteins? So proteins are everything that DNA actually codes for, like the codes meaning the adenine, thymine, cytosine, guanine. So physical things made up of amino acids with elements like Chen, and Chen is carbon, hydrogen, oxygen, nitrogen. These are the 4 elements that are like very extremely essential to living things. And so proteins are made up of amino acids, which are made up of elements like these: carbon, hydrogen, oxygen, and nitrogen. So proteins are in your body, and some examples of like what they do is like they would be what make your eyes the color they are, like brown, blue, black, hazel, whatever. And they also help fight off viruses, which is really essential to understanding our project. So let's look at this little diagram real quick to the left. So functions of proteins. And hopefully this helps you understand how essential proteins are, because they're often overlooked. So proteins are important for enzymes, structure of your body, and like tissues within your body and everything. They transport in blood. They're membrane supporters, which is very essential. They help keep an acid-base balance, fluid balance. They create antibodies, which help fight off viruses, and they're crucial in the synthesis of non-protein compounds and hormones. So when your parents say that you should eat a lot of protein, please take them into consideration. Like your body creates these proteins, but this should help you understand how important protein really is. Okay. So enzymes, which were mentioned on the previous slide, are a special type of protein. They speed up chemical reactions in systems and organisms. Like if you were to eat a banana, an enzyme would help with breaking it down in your body. And these are also known as catalysts. So for those of you who have taken bio or even chemistry, catalysts help speed up reactions and like you may have heard that they lower the activation energy. So you can kind of apply that concept here. It's like the same thing, but in your body. And yeah, so then here are some like an example would be like the banana. And if you look at this image, a substrate moves towards an active site. So these this enzyme is orange, and then the substrates are going towards the active sites, which are these little curves, which they will lock onto. And then the product is released as the enzyme becomes ready for more substrates, if that makes sense. It's making the chemical reaction process much faster. Yeah. So gene modification. So let's do some exercise to sort of replicate gene modification. If you have marshmallow or a couple of erasers or whatever, gene modification is like, it's not in our control. It can either be in our control or it can be the result of a mutation. So if you were to take out a marshmallow and then take another one, but then replace it and then add a marshmallow, even though it's a really small change, and the marshmallows may look the same to you, you took a different marshmallow in your code and the DNA strand is completely different. So think of the marshmallows as sort of the codons that were mentioned earlier, the A, G, C, T, adenine, thymine, cytosine, guanine. And if you could like label them, try it again and see how, okay, I took an A and I took a C and I took a G and I took a T. And then I replaced the T with another G. Like you may think it's a really small change, like, oh, I just changed the letter, but you really changed like the entire protein completely. So it's really essential to the body. Okay, like sort of think of it as like changing a word. Like if you have the word fast, but then you add an E to it, you would think that like you didn't make a huge change. You completely changed the thing from fast to feast. And then if you were to take away feast, if you were to take away the E and the S in feast, you would get fat. And fast and fat are not similar, and feast and fat are not similar in meaning. It's like the big protein change from the small DNA or letter change that you made in this case. So hopefully that helps you understand more like how like significant gene modification can be, even if it's like one codon in an amino acid sequence. Okay. So this brings us to gene engineering. So the process is called genetic engineering, and this will help us sort of go into synthetic biology. But as you can see, like if you look at this photo to the right, you see familiar structure, DNA, but then you see that, oh, sorry about that. You see that a base or from a base pair is being removed, and that you can see like a human hand, so it's being artificially removed. So this is gene engineering. And so if you want to sort of go into more technical aspect of it, you start from a bacterium, which has a plasmid in it. And then the plasmid is, the restriction enzymes are cut in the plasmid, and then a human insulin gene is inserted. And so then the plasmid makes the bacterium genetically modified with this insulin gene, and then it goes into fermentation, purification, and then the insulin is ready for distribution. So gene engineering is used to treat either viruses or diseases and so on. So let's go to the next. So now that you have some more background knowledge on DNA modification, if you could change your DNA to do anything, what would you do? Would you like make yourself more immune to a disease, or like what would you want to do? And if you could design an organism to do anything, what would you do? And these might be scary questions, like, wow, humans really have so much power to manipulate organisms or create something out of just through genetic modification. So it's really something to wrap your head around, because we have really advanced to this point and it is becoming more and more possible. So try to think of some fun responses and what you would do. And really, synthetic biology has no limitations except your imagination. Like you can really come up with anything, and there should be a solution to it. If not now, maybe a couple decades into the future. So it's really interesting. So in the past years, our team has worked on. So this is our fourth year in iGEM as Del Norte. So we've had three projects, and so 3 years ago we worked more with drugs, and then 2 years ago we worked with plastics and the environment, and last year we worked with preeclampsia, which is a pregnancy disorder, and yeah, so it's been a variety of issues, but it's funny because synthetic biology has been effectively applied in all three of these situations. So this year, we're working on lung cancer, which is more similar to preeclampsia in the sense that they're both human, like one is like disorder or diseases, and but lung cancer is completely different in the sense that completely different things are involved. So this year, as those of you who attended yesterday's workshop may know, our iGEM team is using synthetic biology to design a quick and easy diagnostic tool for lung cancer. And yeah, so let's go into it. So through our project, we are also giving awareness for help. So we're like in our workshops, we're talking about prevention, early diagnosis, and trying to destigmatize cancer. So specific to lung cancer, it makes it less scary and helps us find a treatment faster. So we hope other things not only find a solution for early diagnosis, but also help people prevent and sort of destigmatize lung cancer, because it really is an issue that should be talked about more so that people actually help out and help us find a treatment. So here's a little rundown of our project. So Del Norte iGEM, and so we're using VOCs, or volatile organic compounds, in our project. So over 500 different kinds are typically present in human breath, as well as blood and urine. So VOCs, like I said, are present in human breath. And so we are relating this to lung cancer. So how do VOCs, which seem like they're in the breath, relate to lung cancer? But remember, like breath is related to lungs. So many diseases cause the concentrations of specific VOCs in the breath to change in lung cancer. So the VOCs, the concentration or the amount of VOCs in the breath change as a result of lung cancer. So significant changes in around 20 VOCs have been identified as possible indicators of lung cancer. So this means that we can use these VOCs to detect lung cancer using the breath. So how will we detect them? So using synthetic biology, we have designed various plasmids, which were shown earlier, that cause a visible color change when present of a certain VOC. So in other words, the plasmid is designed in the sense that when a VOC is present in the breath, there will be a visible change in color so that whoever is taking the test or whatever understands or knows that, okay, lung cancer is present and this is accurate. By quantifying the concentration of various lung cancer indicators, we hope to provide a non-invasive, which means like not going in surgical or anything, and accurate diagnosis via exhaled breath. So the reason that non-invasive and accurate diagnosis are really, really important is because current treatments are either non-invasive and inaccurate or invasive and accurate. So we're trying to get the best of both worlds and also geared towards early diagnosis, which is really important. Slide, we talked about how prevalent lung cancer really is and how it's becoming a bigger and bigger issue. So these are some examples of past awareness things we have done. So iGEM, like the synthetic biology is part of it, but another huge aspect of iGEM is human practices, which is sort of the aspect, feedback, and helping communities. When we did our project on preeclampsia, we posted some infographics like the symptoms of preeclampsia or mythbusters, like destigmatize preeclampsia and help with like with the symptoms of preeclampsia, would help with either prevention or getting people to get tests done, or for mythbusters, we didn't want people to have misconceptions about preeclampsia. Yeah, so your turn. It's time for think of that you may have left today, or that you will research. And you can pick a topic. You can pick lung cancer, preeclampsia, or you can think of your own. It can be anything related to science, or anything really that you can apply synthetic biology to. And so we'll give you some time to research your topic. Here are some helpful resources. CDC is obviously a really good resource, and www.cancer.org has helped us a lot in our current project. And there are other reputable sources that you can use. And what you're going to do is you're going to come up with a myth that you may have heard or know isn't true, and write down the truth. So I'll give you some time to do that. Oh. So now, using that myth, you need to create an infographic. So you can either draw it by hand, or you can use Canva, which is what we used for our infographics, which is a free graphic design software. And remember that you must cite your sources and add in information. And here's another example to the right. And it has images and text featured equally. It's supported by cited data, and make sure it's accessible. People should be able to get all of their information fast. Like, it shouldn't be like a bajillion words just crammed up in one little post. It should be clear and to the point and really effective. Awesome. So you can share it with us, and we'd love to see it. So thank you so much for attending our workshop today. I know we went through a lot of content really fast, but it's really important that we all understand how important synthetic biology can be in these real life issues, and how lung cancer is becoming a huge problem, and we must address it. And yeah, so thank you for participating in our little activities, and we hope that this was educational and fun for you guys, as it was for us. And feel free to hang back if you have any questions, either about the material, Del Norte iGEM, or just life, high school, anything in general. We're here for you, and thank you so much for attending our workshop.